Okay, so let's do a little overview of where we can find such data and what does this data look like, or what can it be used for. So again, not a long time, because we will spend a little bit more time on some of the practical things so you can try and get started, but we'll do a brief overview. So one of the things that you will find very useful, I think, and intuitive is this American Gut project. The American Gut Project is a citizen science project dedicated to metagenomic sequencing of world citizens that are contributing their microbiome samples to the study of healthy or almost healthy microbiome variability. So the idea is, can we ask people to collect their microbiome samples, skin and gut samples, send them, sequence them, and then we are starting to collect this natural variability. And one of the examples that you will find in the American Gut Microbiome Project, which we will actually try and do in this program, is look at an individual single sample, right? So let's say I contribute my sample. How can I look at my sample? For example, you can see right here, my sample is this first bar compared to the average. Similar diet, so people that are on a similar diet like myself, same gender, similar BMI, age, and some individual, Michael Pollan in this case, so some individual that we can compare. And as you can see, the comparison consists of different colors here that are annotated right here. And for kind of understanding my individual sample, now we can start looking at the most abundant microbes at different levels of taxonomy. So these different levels of taxonomy now can give us some reference that we can try and see what is known about these individual microorganisms or taxa of microorganisms. Down below, you'll see how different sites, so HMP here stands for Human Microbiome Project. So that's where we get the reference, kind of how does this compare to the population? So how can we compare to some of these other sources of information from different body sites, different ages and populations, and the American gut population. So this is a project that I think is fascinating. Provides us an insight into what could be done just kind of to understand what's going on, but also together with this information, you can find metadata. So that was sequencing data. What are the microorganisms that are present? This is an example of metadata. So this is metadata from the Human Microbiome Project. The Human Microbiome Project contains a lot of different variables that are being collected from individuals. And in this one example, we are using a comparison between people that exercise rarely and exercise regularly. So here you can see we can control for certain conditions. For example, they don't have the liver disease. They are considered to be a healthy subset of people. They rarely or maybe regularly supplement with vitamin D. But the biggest difference here is that the exercise is never versus daily, right? So one of the questions that you could ask from such data is, if we control for as much as we know for the different factors, could we find some individuals that are different in one particular way? And then based on that difference, see whether there is or is not a difference in their microbiome. So just an exploratory, simple question. And we'll take a look today at some of the ways that you can start thinking about these questions using this kind of data. Another example that we will look at is how the skin microbiome varies between acne and what's called perioral dermatitis. So a very common condition that a lot of people have either experienced or might continue to experience. The perioral dermatitis is more of a rash, and acne is a more acute condition. And then, of course, we also have areas of healthy skin. So one of the questions could be, how do these microbes essentially interact with the skin itself in inflammatory acne, right? So how do they cause inflammation? And so here in this discussion, we can also download the data, and we'll look today at where this data could be found and try to understand what are some of the differences. So in this case, the difference is represented in this image right here, which is a non-metric dimensionality scaling plot that allows us to see different samples positioned in this two-dimensional space and maybe show us specific grouping based on this annotation, kind of giving us a sense overall whether these samples are different or not. And here we have 3 different samples: adult acne, none, which is healthy control, and perioral dermatitis. Another more complicated study in terms of how it was performed and exactly what was done to develop the data, to kind of prepare the data, is a study that was published in 2015 that spoke about obese gut microbiota induced neurobehavioral changes in the absence of obesity, right? So in this case, the researchers were trying to separate the obesity itself, for example, resulting in some genetic predisposition or maybe in some kind of a diet, from the impact of the actual microbiome or the the composition of microbiota. So could we actually take the microbiota of mice that were fed a high-fat diet, use that microbiota to implant a specific composition into a healthy mouse, and then see whether that led to some neurobehavioral changes? And if you read this publication, you will find that yes, they found an association with anxiety and these kinds of patterns of behavior. But what we can try and do is we can try and take this data and try to understand differences between several different types of data that are available with this publication. Now, of course, these kinds of publications illustrate some of the approaches. They can become more and more complex in nature, and you will find that there is a great interest in trying to understand the functionality, the functional applications, and even develop treatment. For example, here you can see a treatment for Alzheimer's disease that targets the microbiome. So different types of applications of microbiome, obviously going beyond some of the basic research questions, and trying to incorporate the microbial signatures into practical applications, including clinical applications like treatment, or even understanding of the impact on other types of treatments, like, for example, immunotherapy. Today, living in the pandemic, of course, we all might have questions about, well, what is the relationship between the microbiome and COVID-19? And here's an example of a study where the microbial diversity is being studied for long COVID and for susceptibility. So here you will find high-risk groups succumbing to COVID-19. As a result of the pandemic providing us with such a tremendous quantity of data, some researchers decided to collect that data as well, and found some differences between the length to recovery and speed of recovery as well. The microbiome is also used not only in medicine, but also biotechnology and agriculture. For example, microbial fertilizers, MFs, constitute an emerging class of organic fertilizers and consist of dried microbial biomass, for instance, produced on effluents from the food and beverage industry. They have been shown to provide unique benefits, especially in harsh conditions, at the same time with minimal environmental impacts. Metagenomic studies on agricultural applications of microbiome are showing promise, and many have already started using this approach to improve yield and resilience of crops. And so the types of applications of metagenomic sequencing and sequence analysis really continue and go on. And what we'll see is we will see how these types of metagenomic studies focused on the microbiome as a whole are very different from the way microorganisms can be studied in isolation. But just to compare and contrast, here are some examples of microorganism studies in isolation, not as a community, where you can see drug resistance, viral adaptation, or association with a particular phenotype that could be studied once you have the full genome of the microorganism. Okay, so now let's talk a little bit about some of the practical things that we can do to analyze such data. And again, this is not going to be a very detailed overview, but it should provide you with a high-level understanding of some of the practical activities that you should be able to do after completing this training, and how you can continue thinking about some of the research questions that we covered up until now, and try them on different types of data sets. So the first thing that we're going to do is we're going to try and learn this data to pipeline just by understanding the sequence of steps. How do we go from raw data to process data? And then what are some of the outputs, what are some of the visualizations that we can get at the end? So to do that, let's go to the server, which is server.tbio.info. This is a platform that has a lot of the methods implemented in a user-friendly environment. And in addition to that, you will get some demo examples where you don't even need to run any analysis. You just need to understand and visualize some of the data. So please log into this platform. If you don't have a login, for example, you just joined today, you haven't really registered yet, you won't be able to do this together with us now. But if you have created an account on Learn, within 24 hours, you should be receiving your account details. So just to get a sense of how many of you already have an account, could you